When we suggested it, first thing we got a lot of opposition because histidine, an early amino acid, even before life, it's widely believed that it is a latecomer. But from the other point of view, histidine is actually a very simple amino acid that can take leftovers from destroyed nucleotides. So maybe it was an early comer. And with the permission of Dr. Lascano, I'm using his table that he showed yesterday to show that histidine appears in almost all types of enzymes, from very ancient ones to the new ones. Stabilizing the machine is the other job that these oligomers could make. It means coming here to the positions that are still open. And I think stabilizing the machine that makes them is really important. And this is supported by the stabilization of the region within the ribosome, for instance, by protein L36, the one that I told you yesterday that was kicked out. So the summary, it suggested that the genetic code was created by or according to its products, the proteins. Before that, it was not needed. And then the proteins, the genetic code, and the ribosome co-evolved together and optimized for more and more complex life. So that's actually what I said right now. The genetic code co-evolved together with the ribosome as well as with the product, the proteins. Now, RNA enzymes are known to be inefficient. And their nickname is lousy or lazy, depending how good your English is. Nevertheless, in the contemporary world, the ribosome is an amazing chemical apparatus, indicating that nature devised means to turn lousy molecules to efficient apparatus. So we think that the inefficient RNA machine became efficient by the addition of proteins. And actually, in the contemporary world, ribosomes architecture is stabilized by proteins, and some of the ribosome functions utilize ribosomal proteins, but not to do the function, to make sure that the function is done correctly. So here are the ribosomal proteins, the bacterial ribosomal proteins in the small subunit, all of them, in the large subunit, all of them. And you can see that all of them, except for two, I think, have either very long extensions, C and N terminal here and here, or internal extended loops. These penetrate into the structure. I can show here 4 of them, these 4 within the small subunit. They penetrate in, so the small globular parts are on the surface, and the extensions are coming into the structure. So, and we think that we have enough reasons to suggest that they are stabilizing the RNA structure. For instance, here the decoding is happening in the small subunit. A mutation here, far away, but in this extension, does not stop decoding, but it allows mistakes. There are slippages. So we think that this is because the RNA structure around it is not as accurate as without this mutation. So in general, we can describe the structural evolution of the ribosome by core conservation, functional conservation, and shape evolution, growing, because most of the additional proteins that are peripheral, we think that have to do a lot with interactions with the cells. The more complicated the cells, from prokaryotes to eukaryotes, have many more needs to control and to regulate ribosome's function. So what you can see here is the structure of ribosomes in yeast. The gray is not only similar, is identical to bacterial ribosomes, and the red up here is the addition to the structure in yeast. And this brings the molecular weight from 2.5 to 4 millions. You can see everything except for here and there a little hole on the surface. And here are the same pictures are shown here, but I separated the proteins, the additional proteins from additional RNA. Proteins are in cyan, and you can see that most of the additions in the small and in the large subunit are proteins. There is also some addition of RNA, also on the surface, but not as many as proteins. So I want to cite from a paper written in Lorraine Williams' group, comparisons of recent crystal structure of eukaryotic ribosomes with the available structures of bacterial ribosomes showed conservation of the functional core and expansion of the peripheral RNA helices and addition of R proteins, exactly what I told you a minute ago, based on the structure that was determined in Strasbourg by Benjamin et al. So let me show you core conservation, what I mean by that. This is the region, the symmetrical region, the protoribosome, and these are 4 ribosomal proteins in eukaryotes. And I want to show you now the same thing next to the prokaryotes. These are proteins around the region, the symmetrical region, or the protoribosome in prokaryotes. And you can see the prokaryotes look the same, and all are stabilized by proteins. Now I want to talk about another protein that we learned from it a lot about evolution and environmental fitness. And it has 3 names. In eubacteria, it's called L25. In thermophiles, it's called TL5. And in Deinococcus radiodurans, I will show you in a minute, it's called CTC. It's made of 3 domains. The first domain is like the domain in eubacteria. The first and the second together are like the protein in thermophiles, and the three together are those that exist in radiodurans, which is a fantastic bacteria, unbelievable. It can live under super extreme conditions, very hot, very cold, dust, no food, hunger. It is also, it can be irradiated. It also is irradiation resistant. So this is the bacteria. I can talk about how it lives later, maybe only if you want. But in this bacteria, the regulation, it can live under hunger. What does it mean? It means that it can be 4 or 5 days with no food. The cells have a little bit, and it knows not to use it. It's just a fantastic bacteria. So I want to show you this protein CTC that, just to remind you, this is the one that I talked about. Only in Deinococcus it's called CTC. Otherwise, L25 or TL5. So here it is. Here it sits. This is protein CTC, near the bridge that brings in the A site tRNA. Here are the three regions of it, the 3 domains, 1, 2, 3. 1 is like in eubacteria, 1 plus 2 is like in thermophiles, 1 plus 2 plus 3 is CTC. So we think that, not only think, we saw that only one domain stabilizes the position of the bridge that let the first tRNA come in. So this just sits there to stabilize. Together with the second one, it is, we think, that the second domain was brought in by evolution in order to minimize the motions that can happen towards the edge or the outside of the ribosome just because there is more energy. It's thermophile. So this, I think nature made this to protect and to avoid unwanted missions to there. And the third domain, which is only in CTC, is touching the actual age site. And pay attention how it is bound. I'll show you better. This is the way it is bound. Just very, very, very narrow connection, very flexible. So have a look at it with and without A-side accommodation. You can see here, with unbound A-site, we just put it here so you see where it is, and with bound. Here if this actually could not get in because there will be bumps. We just put it here to show. Here the accommodation will be perfect. You can see the whole structure is the same, the whole protein the same except for this part that can open and close. Look how nice. So here it allows binding, and here not. And this just happens with progress of complexity of life in the bacteria time. So let's finish with it. Here is CTC. This is the protection of the bridge. This is the protection of thermal, and this is the regulatory function. So I want to talk now about something else, but also to do with ribosomes. In eukaryotes, there are more and more papers now or evidence that show that the ribosomal proteins may undergo some alterations, and at different conditions. When conditions change, the ribosome changes too, or maybe just can change. So first I want to highlight work done here in Mexico City by Estela Sanchez that took maize, showed that you can grow maize that doesn't grow otherwise if it adds insulin or maize growth factor. And then after she showed that she can grow them, she found fantastic, unbelievable differences in the protein concentration, in ribosomal proteins concentration. Actually, she looked only at the messenger RNA that is responsible for the ribosomal proteins. So she saw upgrade, downgrade, and very few that were the same in the small and in the large subunit. So this shows that conditions can change the ribosome, something that would not be acceptable when I started. So we looked at a messenger for our protein copy number alterations. Now let's look at locations of them. So we are now examining acute inflammation in human in collaboration with Niral Arbel from Singapore. He found that all these genes are either upgraded or downgraded in small and in large subunit, and I really don't want to go one by one. We still don't have more than this, but we looked where these proteins are located in the ribosome. I'll do it real quick. You see the red and the gold are showing those that could be changed by this messenger RNA. Look, all on the surface. We also looked at over or underexpressed protein in human ribosome surface, and we found, as I showed now, that they have something to do with regulation. Now we have some data from breast cancer that are in the protein expression, not only in messenger RNA. And look, even then, there are huge differences. So this shows that ribosomes, not only RNA, but also proteins can change. And I want to cite the work of Isabella Moll. Our results indicate that all cells are equipped with a remarkable toolbox to fine-tune gene expression at the level of translation and emphasize the physiological importance of ribosomal heterogeneity for immediate implementation of environmental information. I couldn't tell it to you several years ago. It's all really new. So, last. Until now, I talked about the prebiotic area, and I want to talk about some medical aspects from the point of view of evolution. So because of the fundamental role played by the ribosomes, many antibiotics target them. Stopping the ribosomes, then stops the life of bacteria. And if we talk about pathogens, it will be bacteria. Actually, almost half of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing ribosomes. What are the antibiotics? The natural antibiotics are the weapons that bacteria from a species, one type of species, use for interfering with cell life of another species. And the question is, how do the tiny antibiotics, where their molecular weight is less than 1,000, can paralyze the giant ribosome? So very clever. Ribosome is a factory, assembly line. Stopping any functional part in the assembly line will stop the whole ribosome. So you can see here the skeleton of the large subunit and of the small subunit, and the bubbles, the balls here, each of them represents a antibiotic family, and all of them are located either in the decoding site or in the bonding, or in the hinging, these hands, or in the tunnel. All of them stop a specific function. And I want to show you a movie that we made based on the first movie and also exists in YouTube. Or maybe until the movie comes, I'll tell you, I didn't put it in YouTube. I have no idea who did it. But it's there and he's dead. Okay, so this is adenine that doesn't let messenger bind to its path, blocks it like a barrier. Also minimizing the flexibility here, but that's it. No messenger, no protein. The second that you will see is tetracycline that doesn't let A site tRNA bind by occupying its position. And the third one is erythromycin, that is actually the first ribosomal antibiotic that went into medical use. Here it is sitting in the tunnel and let only very, very short protein being made. See, that's it. Barrier in the tunnel. And the last one that I'll show you, that's not the last one really, but the last one in the movie, is clindamycin, which from the chemical point of view, it's the cleverest. It just goes into the bond. So have a look here, the bond, pick, that's it. Just sits in. So questions. All antibiotics bind to ribosomal functional sites, I just showed you. But I also showed you, or told you, that ribosomal functional sites are highly conserved. And mandatory for clinical use is the distinction between the patient and the bacteria, the pathogen. We don't want to kill the patient. So if the antibiotics will stop the ribosomes of the patient, will be a problem. Yet, although there is very high conservation, there are subtle differences. Yeah, I wanted to tell you, even if the patient dies when he recovers from his disease, it's not good, right? So we don't want to touch the patient. There are subtle differences. And I want to show you here an example. So these are the tunnel walls. We cut the tunnel perpendicular to its long axis, and these are the walls in gray. Of course, the tunnel is not rectangular. And what you see here inside, each color represents an antibiotic from the macrolide family. There are many of them. All of them block the tunnel. This block excellent, this block very good, this block good, all of them block. And all of them interact with one of the tunnel components called adenine in position 2058. And I want to show you how it works. So this is adenine in position 2058. This is erythromycin. And the affinity here is wonderful. Just chemical affinity, wonderful. Now, please pay attention to the difference between bacteria and us. Here, this point, pay attention. That's all. Bacteria and us. But when there is a G here, there is repulsion, it's too close and no binding. That's all. So the eubacteria are A. Sorry. The eukaryotes are G. That's all. And this is the basis for selectivity, what I showed you just now. And this says that A to G mutation in this position, 2058, according to Ecola, but it's not important, acquires antibiotic resistance. A is in all eubacteria, G exists in all eukaryotes and archaea. This means that A to G mutation increases eukaryotes' fitness. Namely, it's a clever step in evolution. This is what I try to tell you. Something like this. Of course, this is a little place, but that's exactly. So, summarize, A to G at position 2058 is an efficient evolution pathway. Now the last thing I want to tell you about lessons that we learned from the structure of ribosomes of pathogen. So all what I showed you until now was eubacteria, good models to pathogens, but not specificity of pathogens. And it took us 6 years, and now we have crystals of a pathogen. If I'm allowed a personal comment, you see in both, actually we have two of them, but this we already determined the structure of. You see this and that? You know what it is? Somebody can guess? This is, yeah, the eyebrows of our student Zohar. Without her eyebrows, crystals didn't grow, but doesn't matter. What matters is the structure. So I won't show you the whole structure, and I won't talk about many. I just want to show you some take-home lessons from the evolution point of view. So you see here a model, which is a non-pathogenic eubacteria that binds antibiotics very similar, not exact, but very. And here is a pathogen, the cyan. In this part of the picture, they are exactly the same. Actually, they are exactly the same till here. And here there are, it's additional of 10 nucleotides. There are 4,500, so nobody paid attention to these 10 until we found out where they are. Also for you. So here, these 10 are exposed. We are now trying to use such positions for new antibiotics, but what I want to show now is something else. Petrov, it means Williams, Loren Williams, looked at all available structures. And they used ours for eubacteria. These are the red and the blue to eubacteria. And eukaryotes extensions they found here and here. When it says H, it means it's a helix, and each helix has a number. This is what they found additional between bacteria and human, actually not human, they looked at they looked at human. So this is what they found different in these particular places, these additions. Now when we looked at our additions and we compared, we have the same, I just drew it differently, it's the same, more or less the same in these positions, in 10, helix 10, helix 15, helix 16, and I made a nicer picture. So the additions are in two pathogens, the cyan and the purple, compared to the normal U bacteria. Look at it, 2 pathogens, both are extended. And the question that we are now asking is, are pathogens the missing link between U bacteria and higher organisms? This is an open question. So now it's time for me to give some credits. The whole story started with a very intensive collaboration with Max Planck in Berlin. Professor Wittmann was the director. He died 10 years before the structures came, but he was one of the only ones that expected structure, or at least was tolerated. The rest just called me crazy, dreamer, fantasioner. He died 10 years before the structure came out and was replaced by Franceschi and then by Fucini. The real thanks are given to the Weizmann Institute that let me work many years on what was called my dream. Decision was made by Professor Sella, who was a biologist, and was not changed by Harari, who was a physicist. They were advised by the Scientific Advisory Committee. I'm not sure I want to talk about everybody among them, and they also had some advices. But Professor Kendrew determined the structure, one of the first structures, maybe the first, of proteins, myoglobin and hemoglobin, together with Perut. He said a project like this should not be stopped, but will she make it? So this was more or less the Weizmann. So I had 2 groups. I had a group in the Weizmann and a group in Hamburg in Germany, a Max Planck group, that was near the accelerator where we were measuring. And the group in Max Planck was terminated 11 years ago, but the group in Israel is working. So I want to show you, highlight some things, some people, where we measured, and the funding is written here. Ilana is the person that that identified the symmetrical region, and everything came out from there. But for the young female here that already asked me why to do science and so on, I want to tell you about, first of all, you see how many females we have, but I want to tell you that although the society does not encourage, it's possible to be an excellent scientist and also an excellent housewife, mother, and so on. So when I am entertaining you here, the person that runs the group is Dr. Anat Bashan. She is the senior scientist. She has 3 children. She's a fantastic scientist, 3 children, and all of them love her. Ella has only 2 children, but she's also a fantastic scientist. Razu, who was our student, now is a professor in London University. He also has 3 children, but he doesn't count. He's a man, so let's skip him. Let's talk about Tamar. Tamar came 16 years ago for 10 weeks. She loves it. She's still around. Meanwhile, she also has 3 children. But what I want to show you is that I took a picture when she had a birthday, and she baked a cake for her birthday. You may not see the cake, so I will show you. Here it is. And this shows that for my group, ribosomes are considered sweet. Girls, it's possible. I only want to say that to the young female that may hesitate. Shall I stay in science? Shall no? Shall I study? If you love it and you love your family, it's possible. I want to show you how possible it is. Here is my daughter. She is a doctor, but I want to highlight my granddaughter. Even my granddaughter likes me. All my family supported me all the time, and I want to thank them. But she wrote a speech about me, which she read in Paris. And she said, my name is Noah, this granddaughter. As you know, it means me, is a very busy scientist, but she always finds time for me. Yes, it's possible. She even gave me a prize. Now, this is the Nobel medal. That's not what she gave. She gave this one, that for me is as important, or maybe even more. The grandma of the year is Ada Yonat. So, Momentino. So when I asked her which year, every prize has a year. When I asked her which year, she said, which year? You have to reprove yourself every year. And if you fail, I take it off the wall. And so I want to tell you, she gave it to me 9 years ago before the Nobel, and it's still on the wall. So I'm still okay. Oh, please, put off the light. I want to show you now something really nice, but can you put off the light? Constant fight. So actually, this is the only prize that I have to be re-evaluated. And all the other prizes cannot be taken from me. Just only this one, and it's very important for me. A little bit more? Okay, good. That's because I wanted to show you that ribosomes became really popular in Israel. This is a carnival, and he's me, and she is a ribosome. You see, they're dancing, and protein comes out of her. And even young children are ribosomes. And this is what happened to me. So the artist that made it, Michel Kishka, he never met me. Now he met me 5 years later, but at that time. And he is an artist. He looked at the internet and he found, you see the small subunit? And the large one, you see? He took it and he decides that the new antibiotic should be here. So thank you.
Okay, it's late, but we have to have some questions. You can go first.
Good night. First of all, I would like to thank you for your lecture. I enjoyed it very much. And my question is more like a theme to discuss. You speak about the evolution of the genetic code, and you say how important could be that the ribosome be evolved at the same time of the genetic code. But the ribosome is not the only one enzyme that is translating the code. There is also the amino acid tRNA synthetase that also needs to read the code and translate it. And actually they have a very important role because they need to first amino acylate correctly the amino acid, so the ribosome can do also a correct task. So can you relate the components, the amino acid tRNA synthetase, the genetic code, and the ribosome, and maybe try to speculate a little bit about how the evolution of these components can be done?
So now it is, what's the time? What's the time now?
9:05.
Fantastic. Can you sit until 12? Then I finish answering all your questions. So of course, protein biosynthesis is much, much more complicated than just the ribosome. You have to make the correct connection between tRNA and protein. This is the synthetase. I didn't talk about everything to do with protein biosynthesis. I just focused on the part, first of all, that I know better, and second, that we can learn from it something, or at least suggest. I didn't even say that it's clearly like what I said, but we can suggest what happened. In my opinion, first there were proteins, small proteins or oligoproteins, and everything evolved together with it. Maybe you have another suggestion? I'll be happy to hear. But what I think we sure is, or almost sure, is that there was a pocket like this that could make bones. Even
Okay.
this I'm not sure, I'm just trying to show.
Thanks. Good night. What an amazing presentation. And can you use all about this ribosome and protoribosome to give a definition of what is a living organism?
What is, just the last 4 words, can you repeat, please?
If we can use all about the ribosome and the protoribosome to give a definition of what is a living organism or what is life. Or
So I hope that you all sensed that I didn't say that we have already a protoribosome. We have dimers, but we still do not have the real machinery. We still did not show that we can make such a compound. And this is mainly because of technical reasons. We can show binding, we can show dimerization. To show peptide bond formation, only peptide bond is not so simple because it can be also made spontaneously, and we have to distinguish, and the differences in duration are not so large. So we still don't have it, but we are working on it. So once we show it, maybe I will be able to produce or provide you with what you want.
Yes, back there.
Thanks for your presentation. I just want to ask, you mentioned that some extensions in some loops are present in some pathogens, and they also related in the level of complexity in the organisms. I just want to know if it's now the mechanism of how these extensions are acquired in these organisms, because
So, okay, first of all, we have only one structure. So all the rest will be speculation. But this is the situation. But if you think about, the microbiome are thousands of different bacteria, and there are several other thousands all over the world. They are not pathogens. They're all happy. All of a sudden, there are some that are pathogenic. Not so many, but some are. You can ask doctors, they have a list, maybe 2 pages of names. Why did they become pathogens? What was wrong to live in peace and quiet? So we think, or we suggest, that it's worth thinking about the idea that pathogenicity developed with time against the other the other bacteria, and maybe beginning of higher organisms. And maybe this is why I call it the link. Probably not the only link. Maybe it's even not a real link, but it is a progression of the non-pathogenic.
Yes.
Thank you. I have some idea that the ribosome is so efficient that this function to synthesize proteins can be used by some systems like viruses to synthesize proteins to their own benefit. And there are some non-specific interactions that could lead to the synthesis of proteins of a messenger RNA with a non-specific interaction from proteins that seems like initiation factors. Is that correct for all models or some?
Okay, let me clear the question. Ribosome, if it gets messenger RNA, we start and stop, it will do it. Doesn't matter for the ribosome does not ask questions. So if a virus penetrates a cell, the virus takes advantage of the ribosome of the cell. Otherwise, viruses will not multiply themselves. If there is a mistake in the messenger RNA or some clever young people are using non-natural amino acids, the ribosome will take everything that fits into the stereochemistry into the tunnel. So the ribosome is not participating in the mistakes that you alluded to. Those happen at other steps, other components of the same process. Once it comes to the ribosome, if the amino acid is not too large, that is bound to tRNA, it will go. tRNA has to have the right triplet, but if they come in and they are pushed in with 2 out of 3, which is called wobble, most of the time it will take it. So ribosome is efficient, but I'm not sure it's clever.
Now talking about assembly of the ribosome in eukaryotes, the advent of the nucleus and then the nucleoli. What happens in your view in the nucleoli? Why eukaryotes do need nucleoli?
So see,
And also in stressed cells, apparently nucleoli drastically change and you start to require probably chaperones because they migrate into the nucleus. So what is your vision about that?
So about assembly and about the way that ribosomes are being made by the cell, there is a lot of activity now, but it's still not known, surely not in eukaryotes, maybe in bacteria. But the assembly in vitro was shown that it's possible. Nomura showed it in the 50s, that you can get assembled ribosomes from the RNA and the proteins, which is almost as active as the normal one, so it's okay. But in vivo, of course it assembles in vivo, but the stages are not yet clear. There is a lot of progress. I cannot mention everything now, but it's still not clear how it is being done.
Okay, it's late, it's cold, we're losing the audience. I think it's time to call an end to this great session. Thank you so much.